successfully cloned, expressed, purified, and crystallized Arabidopsis sucrose transporters. Participated as a member in a New York Structural Genomics Research Consortium and determined more than 120 protein structure initiative target protein of different organisms, including biomedically relevant pathogens and depositor structural coordinates in the protein data bank, and so on. I am very happy to have such an eminent person in our midst today. I welcome you, sir. I also welcome our Reverend Sister Flora Mary, our Principal Dr. Reverend Sister A.S.T. Lucia Rose, our Deputy Vice Principal Dr. Reverend Sister Kulandai Taras, our HOD Dr. M. Glory, overall Star College Scheme Coordinator and Member Secretary Dr. Reverend Sister Aroke Genesius Alphonse, HODs of various departments, learned professors from various other institutions and from our institutions. Once again, I welcome you all for this wonderful session. Thank you.
Thank you, Dr. Maria Sumadi, for your warm words of welcome. Sir, we are eagerly waiting to hear your sharing your knowledge. Over to you, sir.
Excuse me, Dinah ma'am.
Do you hear me? Or do you all hear me? Yeah. Someone just signed up.
Yes, sir.
Thank you very much. Thank you, Dr. Maria Sumadi, for a very, very detailed introduction. So kind of you. Very good morning to everyone. It's a great pleasure to meeting you all in this great initiative program, even though it's virtual. It's definitely we need this type of initiatives, especially from the colleges from India. I really welcome this type of initiative, and thank you very much for organizing this type of webinar. Thank you very much, Dr. Glory, for your invitation and for this excellent arrangement. And also, I would like to take this opportunity to thank St. Mary's College Principal, Dr. A.S.J. Lucia Rose, Secretary, Sister Flora Mary, Program Convener, again, Dr. Glory, Professor and Head Department of Botany, the Program Coordinator, Dr. A.J. Alphonse, and all other organizing committee members of the Botany Department of St. Mary's College. Thank you all. Last but not the least, I really like to thank Dr. Sonal, Department of Tamil, who introduced me to Dr. Glory and St. Mary's College. Thank you again to everyone, and hope you're all enjoying this webinar. I hope you had a great discussion yesterday regarding the climate changes and global warming. And I'm very sure that we are all responsible in keeping our goal of 1.5 degrees Celsius of global warming. So I'm not going to talk today about the global warming because you all already discussed yesterday. So I'm going to talk a little bit about the climate change and the food security. That is, as you know, from the schedule, I'm going to talk about the climate change and food security solutions, possible food security solution. So at this point, I would like to share my screen. Is it okay to share my screen now? Can anyone?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
All right. Thank you very much. Do you all see my screen? Is it good to go?
Yes, sir.
Yes.
All right. So, as Dr. Sumadi introduced, I'm a structural biologist from Brookhaven National Laboratory, which is one of the 15 US government-funded Department of Energy National Lab. And BNL, we call it as simply BNL, Brookhaven National Lab, is located in New York, which is the northeastern part of the United States. So when you take a closer look, so the New York City, it's also called Manhattan, which is the Manchester of building. It's a big tourism place, as you all know. Manhattan is located here, and the Brookhaven National Lab is located about 150 kilometers from the New York City. It's sitting at the eastern end of the small island called Long Island, and it's surrounded by an Atlantic Ocean, Long Island Sound, and Hudson River. So coming to BNL, so I would say it's contributed so far 7 Nobel Prizes, including 2 from structural biology, and that was nominated and awarded under chemistry division. And it's, of course, it's a 75 years old, you can know it was founded when we got the independence. So this, since you all know about me, I just want to point out my current focus. So basically, I'm currently focusing on COVID-19, especially towards the development of drug, anti-COVID-19 drug. Then second one is called Quantitative Plant Science Initiative. We call simply QSI. And especially in this project, I'm contributing toward the structural biology aspect. The today's talk is primarily lies in this part of the project, So you all know structural biology is an interdisciplinary science. I just recollecting, reminding you, you can ask how structural biology is related to climate changes and food security, So it's interdisciplinary science to study molecular structures of biological molecules such as proteins, nucleic acids, carbohydrates, minerals, so and so on. And it's just nothing but the combination of molecular biology, biochemistry, and biophysics. It's widely used. Structural biology is widely used in medicine, especially towards the drug discovery and agriculture, of course, in industry and in environments. But today we are going to just have a look at the application of structural biology in agriculture. Basically, application of structural biology in agriculture forms the basis of today's my presentation. So this slide gives an overview of my presentation, what I'm going to talk. So first, I'm going to talk how the agriculture, climate change, and humanity needs are connected, how they are related. Of course, we all know. I'm just going to remind you all. And what are the possible solution? Basically, what we are facing is there is an imbalance between these three. And what are the possible solution to fix the imbalance, correct it, how to correct, what are the solutions, current solutions are available, is it enough, or how we can, what is the challenges, how to proceed further, right? So when it comes to this, certainly the project we are developing or we are working on is called Quantitative Plant Science Initiative. Definitely play a small part, not a big challenge, big part, but at least some part it's contributing towards, or it's aiming towards to fix the imbalance, basically, food production, or in other words, biomass production. Within this QSI project, I'm primarily focusing on metal homeostasis in plants, even though we are working on multiple aspects. Today, I'm just stick with this. And then finally, I'll just summarize a few of my results and conclude this presentation. When it comes to, humanity needs, there are three fundamental human needs. We all know: food to eat, clothes to wear, dress, and shelter to sleep. So these three are the fundamental human needs. And these three fundamental human needs are highly depends upon the agriculture, the food production, basically the agriculture. Food, we know, I included here clothes as well, because the cotton fibers and silk fibers, of course, we all know from the agriculture. But what I'm trying to say here is try to avoid or minimize the usage of synthetic polyester and synthetic fibers. So basically, our goal is to minimize the greenhouse gas emission. So when you go for the synthetic fibers, definitely you have to have a industrial waste and the industrial product, and definitely it pollute the system and it increase the greenhouse emission. So basically, when you stick with the natural cloths like cotton fibers and silk fibers, probably, of course, silk is the worms are eating the plants. So that's why I put the cloth as also a primary demand. And then, of course, the shelter, it's the houses, not just the house. Again, here, minimizing the usage of the plastics, synthetic fibers, synthetic materials. Try to use the plant-based or wood-based construction materials. I'm not saying don't use it. I'm trying to say minimize the usage of these synthetic materials. All right. So what I'm trying to say is these three fundamental human needs are highly related to agriculture. Sorry. Yeah. So this, then agriculture is, we all know, it's purely dependent on nature. So if you collapse our natural system, basically the climate changes, then we disrupt our agriculture system, and that will put in a big danger, bring the high demand for our fundamental needs. So basically, humanity needs nature and agriculture, should be balanced. All should be balanced in order to keep our life easier, balanced way. But what we are facing right now and what we are expecting, what's going to happen in the future is the nature, the climate changes. So drastically it's changing. And then the humanity needs the food production, food requirements is increasing. The humanity needs are increasing, not only the food, the other humanity needs I mentioned before. The humanity needs are increasing, the climate changes are increasing, and hence the agriculture products, food and other stuff, other agriculture based products, I would call like a biomass, are decreasing. So if it continues this in this ratio or in this proportion, we are going to be in a very really dangerous situation, right? So in addition, so we are having the population increase, and we are having COVID-19 pandemic. So these are all affecting, put our humanity needs in increasing this one like a balloon, like keep on increasing. And of course we are greenhouse gases emission, and these also change, increase, but these guys is keep on shrinking. So we have to address this. So how to keep these three guys balanced, right? It's very important. It is not only, it's a global issue. We all know it's a global issue. It's a national, international issue. Everyone is responsible for this, each country, and even everyone, each human, all the humans are responsible to, control this, especially the, come to individual, we are wasting a lot of food, we have to minimize the wastage of food. For example, according to UN, there are a particular, specific, significant percentage populations are in hunger, but unfortunately, a lot of, significant percentage people are obese. So basically, we need to balance all those. Anyway, there are a lot of factors. So basically, how to address this? Of course, we know this climate change, so I'm not going to talk. So the bottom line, the fundamental concept is to increase the biomass. So basically through the crops. Of course, we are having animal, but I'm not going to talk about it. So how to increase the biomass by increasing the crop production. And of course, this will help to improve the food production and the other plant-based material, and of course the plant-based energy stuff like bioenergy and bioproducts. So that is our ultimate goal. So basically we have to improve our biomass production. So how to improve? Coming to the food system, so we all know the most important food crops. Of course, we all know rice, wheat, corn, soybean, etc., vegetable, greens, and fruits. These are our main plant-based food systems. So how to improve? What are the solutions? So we are having GMO, genetically modified system. Of course, a lot of genetically modified seeds, to increase food production. And of course, there are technology or GMO-based technology and other technology to make drought resistant, pesticide resistant, herbicide resistant. And the most important technology or important concept which I like is keeping our soils healthy and naturally fertile. There are a lot of researchers are going in these three areas. So here, especially in the case of soil, how to keep fertile, because we are losing our biodiversity because we are using the fertilizers. The fertilizers are killing our microbiome system. So we have to minimize the fertile so you can increase the microbiome, you increase the biodiversity so that it can help reducing the greenhouse gas emission. So there are a lot of researches going on, you all know. So question is it enough? Is it enough to control is it enough to balance the future challenge? Certainly no. According to UN, it's not enough. So we are very behind. We must accelerate or explore the multiple avenues. How to explore, how to accelerate? Of course, we can accelerate, but the problem is the plant biology is basically the basic science in plant biology system, especially at the genomic, functional, and molecular level are missing. Because when it comes to the human system, it's tremendous amount of work, tremendous amount of research are going on around the world. It's going on for long years and years. But unfortunately, plant biology, as it's not that wide, that wide. That's what I'm saying. I'm not saying nothing, not saying it's not going on. It's going on, but what I'm saying it's not enough. So why? Because there are the fundamental, the basic science on plant biology is still missing. So there is a big knowledge gap. So we have to fill that knowledge gap in order to manipulate any system. So we have to somehow we have to understand this biology full and full. Not only just one plant. I'm talking about the most of the crops and most of the energy crops and food crops, all those stuff. Oops. Sorry, there is some delay. Sorry. All right. So this is for one example. Just for I'm just giving one example how behind we are, right? In terms of in terms of genomic and molecular level. For example, we all know that Arabidopsis is the model plant. Right. So many plant biologists or research is going on in Arabidopsis, right? But still in Arabidopsis, the researches with the experimental evidence is only about 30%. It's really not really great thing. There are about 60% are predicted, basically based on this experiment and based on the theory, computational bioinformatics tools. There's 60% of the informations are predicted or like annotated. It's not experimentally proven. And then there is a significant percentage of information is completely missing. We don't know. So imagine this Arabidopsis is a model system. We are very behind in getting this information. Then how we are going to get the information, how we are going to know all other information of other crops, for example, like rice, wheat, all other food crops, right? So I'm giving some of the bioenergy crops, for example. So here, more than 60% of the genomic information is unknown. And same thing, popular, you can see the grade areas are all nothing known. It's not much known. So if you don't know the majority of the genomic information, and of course the molecular function at the enzymatic or the protein function at the molecular level, we can't do much. So we need to understand, we have to know, get this full information in order to manipulate or in order to enhance the biomass production and food production. So this is the our fundamental aim right now. So that's why, sorry, there is some things going on. The slide is not moving faster enough. Sorry. So that's why Department of Energy, US government Department of Energy, funded multiple bioenergy centers. These are all, it's not a one institution, it's like a consortium, multiple research institute joined together and attack a special task, attack the special metabolic system or something to improve the biomass production. And not only there are bioenergy center, there are two different types. One is bioenergy centers, and there is another center called scientific focus area. So US government is funding actually investing a lot of money in these two centers. One is bioenergy center and another one is called scientific focus area. So one of the scientific focus area which we are working on is called the Quantitative Plant Science Initiative. It was founded to address the biomass production under the SFA. We call it as a SFA, scientific focus area. So it is not just, again, one institute, it is like a consortium. So multiple research institute, multiple institute combine together, come together and address particular issues, right? So it's basically what is quantitative plant science initiative. It's nothing. It's very simple. It's basically how to choose, basically how to study the particular system at the genomic level, right? Understand the genomes. So basically, you have bioinformatics tool and phylogenetic tool. There are a lot of computational tools are available to understand the genome, predict the genomes, or in terms of, predicting means predicting the function, or what is the biological role of the enzyme or protein, and then study those enzymes and proteins in vitro, that is the molecular level, understand the biochemistry and study at the molecular level, and understand the protein function of the genes encoded by those genome, basically the proteins encoded by the genes of a particular genome. So once we know the protein function or at the molecular level, so we can definitely, we are very sure that we can go into the system level and alter the system, means that that is the real plant biology, plant level. I'm talking about the plant level. We can change the plant level system, and then if it works, then if it doesn't, then it can come back and then go like a 3 cycle until it works what we are looking for, right? All right, so this is the basically fundamental concept of the quantitative plant science initiative. So basically, again, it forms the QSI forms the foundations for the plant protein function understanding, and it is a multi-disciplinary integrated capability that leverages unique BNL capability to gain fundamental sequence to function understanding needed to improve crops production, especially sustainability and performance. So this involves, as I mentioned, it's a genomics, genetic study, biochemical study, structural study, and then the computationally model the system, complete plant system, and then go into the field work, at the system level function. All right. So the main objective, again, I just want to make it clear, the main objective of the QSI project is to understand the protein functions of all the proteins that are encoded by a specific genome, and then use that information, molecular information, to map or modify the system level function. Right. So there are multiple systems we are working on, but today I'm just going to focus on only the metal regulation or maintenance, specifically iron and zinc. So we all know there are 14 essential elements that must be assimilated from the soil. So, there are macronutrients and micronutrients, but the micronutrients I'm going to talk about is the iron and the zinc is very, very important, because according to the law of minimum, yield is proportional to the amount of most limiting nutrient. It doesn't matter whichever, whatever the nutrient is. So we have to maximizing yield requires balanced plant nutrition, especially the micronutrients. When it comes to soil, you have to look at the concentration of the nutrients and bioavailability, and the soil microbiome that can positively impact the bioavailability. And of course, the micronutrient fertilizers are expensive and only used on high value crops, but our long-term goal is increasing the yield without increasing fertilizer use. That is our long-term goal. So, and also another important goal is, sustainability. So basically do more with less. So basically the benefit, get best out of it. We say, right? Get best out of it, right? Get best out of it like this, like, get the high yield with minimal resources and minimal land use. All right. So we basically, there is a critical knowledge gap in this system, in the metal homeostasis. So we need robust models of how crops respond to nutrients in their environment. So basically, we have the model system continue to provide. It's basically, like Arab does, provide essential foundation for knowledge, but the molecular level details are scanned, and our understanding of how this process impact phenotype in food crops and bioenergy crops certainly is lacking. So, we all, just quickly I touched the iron homeostasis. So it's basically, it involves five steps, basically the iron maintenance in plants involves 5 steps, iron uptake and transport and distribution, and then use of iron in cofactors. So that is the one I'm going to talk about in the metabolism. And then the storage mechanism in seeds, and of course the tight regulation of these 4 processes. All right. So use of the iron in cofactor, basically it's a heme cofactor. So up to 90% of the iron found in leaves in the chloroplast complex as an absolute requirement for iron cofactors. For instance, heme. The heme trafficking and insertion have been described, specifically for cytochrome B and then C-type hemoprotein. But how these hemes are protected from the oxidation, stored and transported by the generalized heme chaperone or heme storage protein has yet to be identified and studied. So that is our basically objective here, immediate objective or immediate goal. So if you understand the molecular mechanism of heme synthesis, transportation, and storage, we can modify the plant at the system level to improve the biomass production. So basically, based on the genomic analysis and also combining and also with the phylogenetic analysis, we have identified three heme binding protein families. Actually, it's all comes under large, protein families, heme binding protein families. Within the heme binding protein family, there are 3 subfamilies. So the first family, which we predicted to contain novel heme chaperone, and definitely it is not characterized. So it is whatever it is, it's based on this sequence level, based on the prediction. So, and the first family is basically a heme chaperone, contained, predicted to have heme chaperone activity, and it is composed of uncharacterized cyanobacterial protein and contains a conserved domain called Duff domain. Right? And then the second family, which is oily in plants and algae, where the Duff domain is fused with the PN-POX domain at the N-terminal. So it's a 2 domain, but it's predicted to bind to heme again. So, and then third family, which is called the Hugsy family, where the Duff domain and the PN-POX domains are basically switched. So here we have the Duff domain at the C-terminal, whereas in this Hugsy family, we have the Duff domain at the N-terminal and PN-POX domain at the C-terminal. So this is the 3 families under heme binding large protein family. These are all the 3 subfamilies we have identified. So our goal is to find out what is the function of these 3 family proteins. It's a family. It's not just one protein. It's a protein family. So each protein has more than 100 members. So first family, just quickly go through, the first family is the Duff domain containing protein family. Right. Right. So it is, so our first question is, we predicted that Duff domain binds to heme. Our question is it really binds to heme? Number one, experimentally we had to prove, right? And then if it binds, how it binds, and then what is the biological function of this protein? These are all our three questions. So in order to answer this question, so one of the protein member, one of the family member, because this protein family has more than 200 members. So you don't have to, in order to speed up the process, just to choose one representative from the family and characterize it, and then we can model the other system, other proteins again. So we chose one from the cyanobacterial homologue, and I just called the Q115 for simplicity. It's a protein. It's a small protein, and so this slide shows general protein production strategy of all QSI target, not just this one. So the following other proteins. I'm just giving the general strategy. So basically we clone our we synthesize the target gene and clone in a specific vector and express them in the E. coli most of the time. If you have any difficulty, we always go for the eukaryotic expression like insect cell, yeast, mammalian cell. But most of them are through we try to express and purify the protein through the E. coli system. So I express in E. coli, basically, purify the protein by different chromatographic techniques. Okay, so the challenge here is, because this is not this one, the cyanobacter, but we are dealing with the plant proteins. So the plant proteins are not easy to produce, because especially when you express in E. coli, the plant protein, it's a heterologous expression, right? So we know. So it's very often tends to misfold, undergo misfolding, and hence it's very difficult to get soluble, well-folded protein. That is one of the challenge we always have in producing the plant proteins in a different expression system, especially in E. coli. But of course, we purify after multiple stuff. So we purify the protein. And then there are three types of characterizations generally we do. First one is the biochemical characterization. Of course, if this is a heme binding protein, so certainly we carried out the heme binding assay for this protein. And then the biophysical characterization is to see the behavior of this protein in solution, how the solution behave in how this protein behave in solution, like different scattering techniques. We use different scattering techniques to find the behavior of this protein in solution. And then the third is the structural characterization. So basically we crystallize the protein, the specific target protein, and then get the X-ray diffraction data from the single crystal, and we use different computational softwares to derive the three-dimensional structure of the protein, right? And not only by X-ray crystallography, we use X-ray crystallography, as well as now the recent technique, you all might know. It's another technique called the cryo-electron microscopy, which is also another powerful technique. In fact, of course, it's a very powerful technique to study the complex system. If you have multiple proteins, if you want to study the protein-protein interaction complex system, the cryo-electron microscopy is another technique we use. So these are all part of the structural biology techniques. All right, so of course, coming to this protein, so we purified and we characterized it. Of course, as predicted, it binds to heme with a KD of binding constant of 1.5 micromolar, which is good, which is similar to the heme oxidases, but it cannot degrade heme. It binds to heme, but it cannot degrade. It didn't degrade the heme. So that suggests that this protein could be function either storage protein or a heme chaperone. All right, so, and then of course, now we purified the protein, we characterize the protein in vitro, so we know it function, it binds to heme. So our next question is, in order to manipulate the system, we need to know the molecular level information, right? So that is very important. In order to completely manipulate, genetically manipulate, we need to know the system, we need to know the information at the molecular level. So we crystallize the protein, as I mentioned. So we basically, different techniques are available. So crystallize the protein, it's a single crystal, and we have a unique, we have a very, very world-class facility. It's called National Synchrotron Light Source. So it's basically nothing but you have the high energy X-ray beam source. So you have the high energy X-ray beam source. So basically you produce the X-ray at a very high energy and pass the X-ray beam to the crystals and collect the X-ray diffraction data within at a very, very faster speed, at a faster rate. So this is it's one of the world-class facility. We use this facility to collect the X-ray diffraction data, and this picture is just to show the what actually exactly how it looks like in the hutch. This is one of the hutch. So we have like more than 50 hutches in the synchrotron ring. So it is a big facility. This is one such station. It's called hutch station. So we mount the crystal here, and then basically the crystal always should be in the cryo stream, and we pass the X-ray and collect the diffraction pattern. We have high speed detector to get the pattern, and we have the multiple computational softwares are available to determine the derive the 3 dimension of the protein. So this slide or this picture is a cartoon representation of the structure of the Q115, one of the target protein. It's actually, there are 4 subunits in the crystal asymmetric unit. The 4 subunits you can see in the 4 different color. But when it comes to the molecular interaction, there are 2 types of intermolecular or the interaction or the subunit interaction. 2 types of subunit type interaction. One is through the beta strand and another one is through the metal ion. So what is the interesting feature here? So from the sequence analysis, we predicted that it binds to heme, but we didn't expect to have a metal binding site one. Okay, so in the crystal, in the protein, we have the metal binding. So that we came to know only after determining the structure, which is really new. Okay, so this we identified this metal ion as a zinc. So the zinc is basically forms a tetrahedral coordination, and it is binds at the basically dimer interface. It's one of the subunit interaction, 3 histidine and 1 glutamate from the another subunit. Okay, so it forms basically, it has a metal ion. Okay, so this is the native, so we determine the native, but of course we need to map the binding site, right? Where how heme binds to this protein, what are the heme binding residues. So in order to get the heme binding site, so we determine the we co-crystallize, we take the protein and add the heme and incubate it, and then again set up crystallization, undergo the crystallization process, and we get the crystal, and we collected X-ray diffraction data, and we determined the structure, and here is the structure So the structure basically, it forms a dimer. The two subunits are here. It's a cartoon representation of two subunit, one in green and then aqua blue, marine blue, and the heme is binds at the dimer interface, and there are one zinc which corresponds to one subunit, and there is another zinc corresponds to this subunit, but the heme is binds at the dimer interface, which is, you can see very clearly here. It is like basically the heme is in between these two zinc ions. So this zinc ion is coordinating to this amino acid. Especially, very important to note that this histidine of one subunit coordinating to this zinc, as well as this histidine is coordinating with the ion of the heme. And same thing in this side, histidine, zinc, and then basically the zinc to the other amino acid of the different subunits. So basically, zinc, histidine, ion of the heme. So the heme is saddled between these two zinc ion, which in our knowledge, this is the novel feature. We never come across this type of unique feature. It is very very exciting. It very exciting to see this type of architecture, this type of arrangement. So you can just quickly, I'll show you in the three-dimensional space. Let's see whether, yes, you can see the heme, which is in the salmon color, red color. The stick model is at the dimer interface, and you can see the axial histidines basically coordinating to the heme, ion of the heme on one side, and it is very important, another interaction. The propionate moiety of the heme is making salt bridge with the arginine. So there are two types of interaction. One is the metal binding interaction, and another one is the heme binding, basically the arginine, the salt bridge. So of course, we wanted to see whether these heme binding residues, including the metal binding residues, are real or not. So we mutated, we carried out the site-directed mutagenesis experiment, and we checked the heme binding. You can see very clearly this is a wild type, and it binds to heme, and these are all the mutants. You can see clearly reduced heme binding. Basically, the mutations are, mutating these heme binding residue and metal binding residues to an alanine. Okay, so when you mutate this residue, for example, histidine 79, so histidine 79 decreased activity, and then the arginine 90, we had decreased activity, and we mutate the double mutant, this histidine and arginine. Actually, we expected complete loss, but we didn't. But there is another reason, for it. Maybe I can show it in the next slide. There are two important features we observed from the mutagenesis experiment. Number one, the histidine 16 and histidine 21. Basically, these two histidine, when we mutate these two histidines, we completely lost the heme binding activity. So basically, complete loss of the heme binding activity. This suggests that the metal binding is essential for the metal coordination is essential for the heme binding. Right, and of course, there is another feature is reduced heme binding for most of the mutants, as we expected. All right. So there is one thing, we expected some sort of complete loss of binding for the two residue, which is histidine 79 and then arginine, but we didn't get the complete loss because there is a conformational flexibility in the alpha helices. When we mutated those two key residues, arginine 90 and the histidine 79, there are two other residues which are, the helices at the surface undergo conformational change. it's easy to understand by modeling. We proved, or we propose that, due to the conformational flexibility, there are two other residues like lysine 87 and aspartate 77, which is close by, or compensating these residue. So that's why we didn't get the complete loss of activity, but still we expect we do have the reduced binding affinity. So what basically what are the key observation we observed from this structure? One, from this study, one. So this target protein associates as a dimer when it binds to heme, and heme is binding at the dimer interface, more specifically between the helices, alpha helices. And heme is saddled by the zinc ions, which is the unique feature of this family. And heme is highly secured. It's compact. It's beautifully located in the middle of the interface and protected from oxidation, any type of oxidation by the axial histidine. So it binds to heme, but it cannot degrade heme, suggesting that it functions either as a heme storage or heme chaperone. So loss of heme binding due to double mutation of the metal coordinating sites clearly indicate that the metal ion is required for binding. Okay, so how it recognize, how this protein recognize the heme? So basically, in the presence of heme, arginine 90, those two arginine recognize the propionate moiety of the heme, and it acts like an anchor, so it recognize and bring the heme closer, and then the metal ion position this histidine 79 so that it catch the ion in the heme. So that is our mechanism we proposed for the heme binding. So with this, I'm moving on to the next family. I'll try to move I'm moving little faster. Can I know how long I have now, Madam, Dr. Glory, Madam? How long I have still?
You can continue, sir. Since the session started at 9:20, you can make this of 20 more minutes, sir.
All right. All right.
You
okay.
can
Okay.
proceed.
Yeah.
Very interesting, sir. Though we are not up to the mark, but your session is very interesting for us.
Thank you. Thank you. I'll try to slow down. Maybe I can make it a little bit slower. Okay. Thank you.
No. It's very interesting. You can go fast because it's so purely molecular based. Yeah. No, that's why I say.
All right. So the next family, which is the second subfamily, it is also called glutamyl tRNA binding protein. It's related. It's closely related to glutamyl tRNA binding protein, but it is completely uncharacterized, and it has a two domain. So the DUF domain is at the C-terminal, and then the PN-POX domain at the N-terminal. The PN-POX domain is also, it has a split barrel domain, and it's oxidoreductase domain. And then of course we know this DUF domain binds to heme. So it's a heme binding domain, and it has the oxidoreductase domain. And of course we selected three targets, three proteins, two from poplar, from two different locations, localized in plastid and chloroplast, in the cytosol. So there are two same family, but same organism, but two different location. So there are two targets from poplar and one from sorghum. So just we selected three because even one fails, maybe other can help. Just make use of we are not sure whether if you choose one, whether we do not know whether it works or not, because it's a higher plant system. So definitely it's good to go with multiple targets, so maybe one can work. Right. So yes, we took three, but definitely one of them worked very well. So all these three genes, as as before, as I explained before, same methodology in, synthetic gene cloned in the expression vector, expressed, purified, and then characterized this protein. So what is the difference from the between the previous one and this family? This protein binds to heme as predicted, right? At different, of course, this is the heme binding assay curve. So, at different pHs, it binds. Of course, at the neutral pH, at pH 7.4, it binds at the maximum level. It's not only binds, it's another feature is it degrades the heme. So basically it binds and degrades the heme. Degrade means it has a oxidase activity, oxygenase activity. So basically it binds and converts the heme into a bilirubin or some other product. So it binds and degrades. This is the function of the in vitro study, in vitro function of this protein. So as predicted, it binds to heme and also it degrades the heme. All right, of course, to understand the molecular level, so we crystallized this protein, and same thing, we collected X-ray diffraction data and determined the three-dimensional structure. So here is the structure of this protein. Yeah, all right. So, as I mentioned before, it's a two-domain protein. There is one domain at the, sorry, somehow the pointer is not working. All right, it's okay. So there are two domains. So one is internal domain, and then the C-terminal domain. The internal domain is oxidoreductase domain. I hope you all see my pointer. And then this is the C-terminal domain, heme binding domain. So oxidoreductase domain, internal oxidoreductase domain, and then the C-terminal heme binding domain of one subunit, which is Marian below, and then there are another subunit which is shown in the green color. So it forms a nice dimer. The dimer interface is through the oxidoreductase domain or the PN-POX domain. So here is the dimer interface. Dimer interface is here, but the heme binding domains are free from one another. So there is no interaction between the heme binding domain. All right. So this is good. All right. So what is the surprise? So the surprise is, as predicted, yes. Okay. What the surprise is, we identified a metal ion at the dimer interface. So at the dimer interface, there is two metal ion. Basically, we identified nickel ion, which is one here. This corresponds to this subunit, and there is another nickel, which is shown in the green color sphere, which corresponds to this subunit. So basically, there are two nickel ion at the dimer interface. In addition, there is one nickel ion at the heme binding domain, which is exposed to solvent. Basically, it's completely exposed to solvent. And similarly, here there is another nickel ion. So as a whole, there are 4 nickel ions. So we couldn't trace out at the sequence level. So we came to know only after the structure determination. Okay, so the new concept, the new thing is, it's a surprise, the presence of metal ion especially at the dimer interface, at the oxidoreductase domain. All right, and then this is the close-up view of the nickel coordination. I'm just showing one of them. So you can see the nickel forms a very nice octahedral geometry, and there are 3 coordinating residues from one subunit, and then there are 2 from another subunit, and then the 6th coordination is from the water molecule. All right. Oops. So here is the static picture, just summarize. Dimer, and there are 2 domains per subunit. One is internal domain, C-terminal heme binding domain, internal oxidoreductase domain, and 1 metal ion per domain. One is here, and this is one is in the heme binding domain, and this is exposed to solvent, but this is completely at the dimer interface. Coming to the coordination sites of the exposed nickel ion to histidine from the protein, and there are 4 water molecules. It's purely exposed to solvent. And here is the coordination of the coordination geometry of the nickel ion located at the dimer interface. All right, so we determined the native structure. From the native structure, the surprise is the presence of metal ion. Then, of course, our question is how it binds to heme. What are the heme binding residues? So, and then basically we found, you know, the heme is binds at the in between the oxidoreductase domain, not at the heme binding domain, right? As you know, basically in the previous family, the heme is binds at the heme binding domain, but in this case, very interestingly, that it binds at the dimer interface, basically in between the oxidoreductase domain, and you have the metal ion, metal binding sites are here. Maybe I'll see whether I can show these structures. Hold on, 3D structure, yes. All right, so, all right, okay. So you can see the one subunit is shown in blue color, and there is another subunit is in yellow color. Heme, the heme is binds in between the oxidoreductase domain of these 2 subunits, but the heme binding domains are here. It's not interacting with the heme at all. Very surprising, very interesting, right? And then, of course, the metal ion is here. But of course, we do not know what is the role of metal ion here. We are still investigating. We are doing a lot of mutagenesis experiment and all those stuff. All right. So coming to the so heme binding residues, as we saw in the previous family. So there are set of heme binding residue which are shown in this stick model. Basically, it's mostly hydrophobic. It's not like really like a hydrophilic. It's mostly through hydrophobic interaction, and the heme, the ion of the heme is coordinating to an azide molecule which we added during the purification and crystallization process. So it is the ion basically is not coordinating to any of the protein residues. Interesting. And it has something, maybe it's a, you know, azide molecule. Maybe there might be a water molecule. It was replaced by a azide molecule. So the coordination is a little different. But of course, we are doing the mutagenesis, you know, experiment, site-directed mutagenesis experiment to understand the complete mechanism. All right. So to summarize this study, these families of protein. Of course, the 3rd family, we are still working on it. So we just purify, we are just, you know, characterizing the protein. There are still a lot to go. So I'm not presenting that one. So based on these 2 family, I just want to summarize these. So we predicted 3 conserved heme binding protein families using bioinformatics tool, confirmed and demonstrated the heme binding and degradation properties of predicted heme binding proteins using biochemical in vitro studies. Determination of crystal structures of cyanobacterial homologue in the presence and absence of heme revealed unprecedented features. Basically, in the presence of heme, a protein forms a dimer and saddled by 2 zinc ions in a zinc histidine heme configuration from each monomer, which is the unique feature. By analogy with the protective axial histidine found in bacterial heme transporter, it can be proposed that the zinc saddle found here protects heme from oxidation. Hence, this protein family could act as a heme chaperone. And of course, the crystal structure determination of Poplar revealed new metal binding and heme binding sites, and probably the presence of metal binding sites in between the redox domain of the dimer is basically, it's little unique or novel. We didn't detect at the sequence level. We didn't see in the other structures. So the conclusion with this, in this family, study from this family, so the discovery of the family of novel heme oxidase, that's from Poplar basically, and the putative heme chaperone, this duff domain with this unique heme binding pattern provides new molecular and genomic insights into the evolution of heme regulation in photosynthetic organisms. So basically, we are doing a lot of phenotyping since this project has been started last year. So we are in the third stage. Basically, we are in the fourth stage. Basically, genotyping, and there are a lot of studies are going on. So my collaborators are basically our team members are working on it. So with this, I'm going quickly explain the another system, not much, just few slides, which is basically deals about the zinc ion. So what we saw in the previous was like a met ion. So this system is a zinc. So our question is how zinc dependent proteins loaded with the zinc. Of course, I'm skipping a lot of stuff. So the importance of zinc, you all know. So the fundamental question here is how zinc dependent proteins are getting the zinc, right? There are a lot of zinc dependent protein. There are a lot of functions, or it's carrying out different function. But how this protein getting zinc in the plant system? So of course, from the root, so it transport through the membrane, it come into the cytosol. It's a labeled zinc pole, and then this from the zinc pole, it goes into the zinc dependent protein. So I'm just going one couple of slides, just one case. For example, there is a protein called aminopeptidase, MAP, methionine aminopeptidase, which plays an important role in the protein translation. Basically, the ribosome, initiating in the translation process. That's called methionine aminopeptidase. So basically, that requires metal ion, specifically zinc, for its activity. So how that map, or how this methionine aminopeptidase get zinc, just touch that part. It's a very interesting stuff. So basically, same thing, using phylogenetic and bioinformatics studies, we have identified a big family which has about 40,000 members. It's completely uncharacterized. It's like from the genomic scale, I'm saying, different. So it's a big family, and it is completely uncharacterized. And it is like we call it as a COB-W. It's a cobalamin biosynthesis protein, but we do not know the exact function we predicted. So basically, it is related to GTP-dependent nickel transferases. So it is a GTP-dependent or GTP-dependent hydrolase, or we say it like a GTP-dependent metallochaperone, or it is a GTPase action. Basically, we found that it has a GTPase action. A GTPase means it converts the GTP into GDP, and during that process, it releases the zinc ion, and that zinc ion is transferred to the aminopeptidase, MAP1 protein, methyl aminopeptidase, and then it carry out its function. It converts the peptide bond. It cleaves the peptide bond of the internal methionine of the polyprotein. So basically, we did some, again, same thing. I didn't repeat all those things. Same strategy. We purified, expressed the protein, purified the protein, and carried out the biochemical assay. So basically, what we biochemically characterized. We characterize that this protein has a GTPase action. GTPase means, it converts the GTP into GDP. And of course, it requires some metal ion, magnesium ion. And then what we found is, interesting feature is, we found that, of course, I'm skipping this other type of studies. So what we found was this GMC, or GTP dependent metal transfer. So basically it's a GMC, we call this a GMC or GTPase. So it converts, it hydrolyzes the GTP, and during that process, it releases the cofactor. It's basically a zinc ion. And this zinc ion, in the presence of cofactor, this zinc ion is transferred to a methionine aminopeptidase, right? So, and then this aminopeptidase carry out its function. So basically, we did some experiments. So we took the GTPase, and that's a GTPase enzyme, and then we added MAP. There is no MAP1 activity. MAP1 activity is a peptidase action. There is no peptidase action. And then we added GTP, right? We added GTP to the same thing. So basically, we had GTPase and then the MAP, and we added GTP, and basically it worked. So means this enzyme, the GTPase, and this MAP, work together. Basically, the GTP converts the GTP into GDP and release a zinc ion, and this zinc ion is transferred to the MAP1, and this MAP1 carried out its enzymatic action. So GTPase and release the zinc, and that zinc ion is used for the MAP1 activity. So basically, this GMC acts as a chaperone. It's a metal chaperone. So it provides the metal ion to the client partner, which is the MAP1. So we just, I'm not going in detail. We did a lot of yeast hybrid and a lot of mutagenesis experiment, and we derived some sort of molecular mechanism how this metal transference is happening. I've just shortened this whole study. So this is the complex of the GMC, that is the GTPase, and then this is the MAP1, how these two proteins come together, and basically the GTPase action is happening here and release the zinc, and here is the peptidase action is happening through this active site. So I'm not going in detail. So there are a lot of still some more experiments needs to be done. So and also it's going to get published soon. This is a very interesting system. It's going on. We are exploring in different system, like higher plant, like poplar, sorghum, and also in rice, and all the system. And of course, we are transforming this information into the phenotype and then the plant level. All right. So with this, I would like to acknowledge my team, QSI team member. We have, Kristen is the overall manager of this team. Our postdocs, Nicholas, Cassandra, Marian, Hesla, and there are other scientists within our QSI team, Mong, Tim, and Aditi, Chin. And our funding agency is primarily by US Government Department of Energy, and these are all our collaborating institutions. This is in New York, and this is in Chicago, and this is in Madison. Just before finish, I just want to show a couple of my slides. You might ask, really, is this type of study works? Right? It's a question, right? Yes, it worked. Even I'm just going to show one example from my colleague. So he is a CJ Lou. He's my colleague. He has his own group. Basically, he's working on the plant cell wall. plant cell wall and lignin biosynthesis. So what his focus is to understand how phenylpropanoids and lignins are synthesized, deposited, and assembled in plant cell wall, right? So first understand the system fully, then manipulate the lignin biosynthetic process to improve the woody biomass production, basically biomass processability. I like this one. That's why I gave one example here. So what basically it is, so he found, I'm summarizing here. It's a big project. It's a long-term project. He was working in this project for more than 10 years. But he came, recently got really, I just want to show one example. Is this type of work really works, right? So he found, an enzyme called MOMT. This is a very important enzyme, play a key role in the lignin biosynthesis, right? He determined the structure, and he get the information at the molecular level, and then he modified the enzyme. He modified the enzyme based on the biochemical and in vitro data, and engineered the enzyme and transformed into the plant level. So what he did was, so basically this enzyme is important in the lignin biosynthesis, right? So we change the lignin biosynthetic process. Basically, you all know the cell wall has the, highly recalcitrant lignins, right? It's very, very difficult to digest the cell wall, right? So in order to release the free sugars for the bioenergy product like ethanol, right? We need to digest the cell wall and release the sugar, and that sugar can be converted into ethanol. All right, so that is the whole process, and I'm simplifying this whole process. So basically, by altering this enzymatic function, he loosened basically same biomass, same cell wall structure, but he loosened the synthetic cell wall, loosened the cell wall architecture, and hence the digestibility was very easy, and hence the release of sugar is much easier, and hence he produced more than 40% of expected yield of ethanol. For example, here in this plot I'm showing is a control, and this is the engineered MOMT enzyme. So it's in plant. So it is basically what you're seeing is a 9 months old plant. So he took the stem and put in the industry. So basically it's a patented study. So he found this engineered one produce 40% increase in the ethanol production. So yes, indeed it worked. This study, I'm just giving one example. There are other, even there is another, my colleague working in the lipid biochemistry. He also same thing, increasing the oil content in plants. So basically, without altering the physiology of the plants, that is very important. Without compromising anything, he made especially CJ. So he made this cell wall structure. He loosened the cell wall structure, and basically the stems, the wood can be easily digestible, and then get the release the ethanol easily, and release the sugars, and then ultimately it was converted into ethanol production. So what I'm trying to say is, yes, this type of study is working. There are a lot of researchers are doing, and it is working, and hoping, we started 2 years back. So hoping we are also doing a lot of greenhouse, greenhouse, a lot of field work. hoping to have some more, real type of biomass production soon. All right, there is another system, but it's okay. So with this, I really would like to thank you. Thank you for your attention. It's a great pleasure meeting you all, and I'm happy to take any questions, please.
Thank you, sir, for making this section a really interesting and erudite section, and will enhance our knowledge in gene binding proteins from bioenergy crops. Dear participants, post your queries in the chat box. Otherwise, unmute your mic, ask your queries to the resource person.
Yeah, please feel free to ask, don't hesitate. But when you unmute and just to introduce yourself, please, you can say your name and your institution, and then you can feel free to ask any type of question. I'll try to answer or address your questions.
Hello, sir. I am Dr. Glory. My question is very simple. It seems actually that all the manipulation process you are doing at a protein level, no? You are studying about this metal binding site, and by using mutant and all. Okay, that. How was it be inherited to the next generation? You will go back to reverse transcriptase, just like you will go ahead?
Yeah, so first, basically, we had to do phenotyping work. First we had to do some sort of phenotyping at the plant level, and then, yes, we can go back to the RNA level. Not only RNA level, you can go different ways. So we go through the nucleotide level, gene level. So we go back to the gene and make the gene, vary the gene. Basically, we manipulate the gene and then come back to the function. So we go back to the gene level. Yes.
Okay, sir. Why can't you proceed from the gene level by altering the gene sequence so that the binding site as such can be manipulated, so that
Yes.
the application will be more easy,
Right,
no, sir?
but we do not. Yes. Definitely, it's a good question, but we do not know without, we had to know the protein level, right? So we may not know, we had to make sure, right? So when you go from the gene analysis, so when you analyze the gene at the sequence level, it's predict a lot of other stuff. So what we are saying is just the conclusive part, right? It's a prediction. There you always have a lot of prediction, but we have to experimentally prove. But in order to prove at that point, we need to work at the protein level, because the gene level is not that easy to work. So of course, ultimately we need to get the protein level information, right? Then only you can go back, because at the gene, I got your question, but definitely it's a good question, but it is very, very hard to do that. Right, you all know.
Then again, sir, you are working with the protein of proteins. I mean, oxidoreductase or cytochrome isolated from chloroplast. Which you are working with a protein isolated from chloroplast, plants of chloroplast protein, all these oxidoreductase associated with the electron transport chain in the light reaction.
Right. So there are two, as I think I mentioned this. So this localized in two different locations. There are two genes we selected, one from the cytosol and then one from the chloroplast. Yes. Answered. I think you wanted to know where it is localized, right? Yeah. So one of them is localized in chloroplast. Yes, ma'am. Yeah, please proceed.
Actually, my question is, we are familiar with various ion binding protein taking part in photosynthetic process, particularly of light reaction. And my question is, even otherwise, the soil is not deficient of iron, particularly in the Earth system. Then also there are some iron chelators, and availability of iron is also rich in the soil. Though chelators are blocking in somehow rare plant system show iron deficiency symptoms. Then what's the meaning of working this making manipulation and homeostasis of the plant? There are so many climatic system and minerals in the earth, how deficient of various other ions, other micronutrients. But as far
Right.
as iron, Fe is concerned, it is not insufficient to the plant all over the world. Then what's the sense of, my question, what's the sense of doing all experiment related to iron homeostasis?
Right. Right. So definitely, I got your question. So we are, I think I mentioned before, so we are facing multi-environmental situation, and we are looking at the genomic scale. We are thinking, the iron deficient. So I didn't show there are some of the soil related or iron deficient knockout experiment, all those things. But definitely the reality is not we are thinking. The iron deficient, we are thinking that it's not very important, but definitely that is not the case. And not, it is not, it's again, it's not only the iron deficient. So this type of system can be extrapolated to a different system, like other type of system. So whatever we are thinking, having enough information is not enough. That is that's what I mentioned before.
Thank
Maybe
you so much,
I would
sir.
have
Thank you so
bring,
much.
yeah, I would have bring some. I do have those information. Iron deficient. We are doing a lot of agro material where we have iron deficient and zinc deficient plants, and then knockout gene, all those things. But I didn't bring this here because we still needs to do a lot of some confirmatory test. And moreover, I try to reduce the complexity. I make it as simple as possible. Definitely, it's a very
These are
good question.
maybe
Yeah.
of the question may not be relevant, just like you thought about this lignin deposition. Of course, actually, when we lightened or softening the lignin, that lignin only giving all sort of rigidity and giving shape to the cell and making the all sort of mechanical support to the plant. When any work softening the lignin through gene manipulation, is whether it will work out in natural uh, complexity. My question is, when softening by manipulating the gene, controlling the lignin deposition, thereby, of course, your scope will be attained, softening of lignin will taking place. When softening of lignin taking place, my question is, it will affect all the complexity of orderly complexity of plant life.
Yeah, definitely. It's a good question. That's what I think I mentioned that figure. So definitely, it's a good question. But fortunately, it didn't affect anything. That's what in that picture he explained, without affecting. So basically, the softening, I use the word. It's a general word. It's not really the softening the lignin. So there is it's a phenyl propanoid complex. So basically, there is a linkage connecting this lignin. So basically, he just knock out those, but definitely it didn't alter any of the, it's maintained the strength in the and it is very interesting. That's what I brought that one here. experimentally, he showed, maybe you might have seen in that slide, the plants are looking same healthy, and it is 9 months old. It's everything is beautiful with respect to the control. So what he found was that ultimately, take the stem and, crush it and, undergo the digestion process and release the sugar and then to the, final to the ethanol. So that is the important part. But what he measured, he did a lot of, other control, he make sure structure and make sure the, that is very important. This is also another important aspect you brought. Actually, they are working in rice as well. Now, actually, what is the important part here is without affecting the rice production, what he is trying to do is, the straw, right? So after the rice, we have the straw. So from the straw, we can, because, it's a recalcitrant, we know cell wall architecture, right? So how to get the ethanol from the straw in a cheap or in an economic way. So that's why the whole process is, cost effective, right? So making, less price, less price and get the more ethanol. So bioenergy product. So it is working in the rice too. So he is, as you mentioned, he controlled all the, not controlled, he studied all other parameters. So basically, that's what its good thing is, it didn't affect any of the other structural features of the plant. So he had the control. It's like a control only. It's very specific. That's why that's why it's important to go at the protein function, protein level, and then go back to the gene. That is the one specific example I can say. That another important thing, as I mentioned, he's working on the rice too. I think he's getting very, very exciting results. So soon it will be published. Yeah, definitely. It is always, when we manipulate the system, always it affects the other, we always control the other parameters. It's always there. Yeah. But this one, our goal is, that's what our goal, without disturbing the complete architecture. That is very, very important, right? So we don't want to disturb the production, especially for example, crops like rice, wheat. We don't want to disturb the production, but at the same time we try to make use of these straws and other stuff, how to convert those stuff into usable, more usable or the economic way. Am I answered? I know it
Thank
is
you,
very,
sir. Yes,
very
sir, I got it. I
yeah.
got it. Got it, sir. Thank you for your detailed explanation for me. One more, my discussion only, it is not
Sure.
making, but instead
Yeah.
of doing gene manipulation associated with lignin and crop plants, is it because as far as biomass is concerned for production of ethanol or any other things, it is better to manipulate, already manipulation done, better to manipulate more microorganism capable of producing very effective lignocellulolytic enzymes, which is easy instead of manipulating the crop plants at crop
Yeah,
level.
yeah, yeah,
Is
yeah,
it because it is a fermentation products? So fermentation
yeah,
products, of course,
yep,
we can't be
yep.
strengthened. My question, this is not my question, it's my just discussion and sharing. Why can't we make more research on making more microorganism which is lignocellulolytic at the time of fermentation? So
Definitely.
what?
Yeah, definitely, definitely. There are bio, as I mentioned, I'm just showing one, I throwed one example, but as I mentioned, there are bioenergy centers, right? They are doing it. They are doing it. They are developing the microbial system to the cellular lighting. Number one, they are, yes. Your question, they are doing, people are working on it. Even our other bioenergy centers are doing it. Number one. Number two, and again, the microbial system. Number two, again the enzyme cellulases. Actually, last few years back, in fact, I was involved in this digestion, digestion process, the cellulases. In fact, we modified some of the cellulases and then the microbial system together, right? So there are multiple avenues. That's what I'm saying. So we have to explore all different ways, right? It's not just one route or just one task. It's definitely it's a multitask, multiple avenues, multiple, it's wide things. So what is the bottom line? As I mentioned, is single route may not give a solution for us. We have to explore all different routes. That's our fundamental concept. Yeah, definitely. Yeah, we can, and the people are working on it. Our bioenergy centers, there are few bioenergy centers are working in the microbiome system. Yeah, definitely.
Thank you so much, sir. Participants, thank you. One question, other participant, please ask your question.
Yeah, boy, everybody's silent. Maybe in the morning time, busy with the morning class. Sorry. Or maybe understood fully or don't understand anything. There are two ways, right? Maybe don't not
They would
clear enough.
they would have understood, but this is at molecular level, no. That one
Yeah,
who
later.
is highly engaged with molecular genetics could raise question. But that's what so much of discussion. Some scientists could
Yeah.
raise questions like this. That's what the reason. But as participants were so attentive till the end of your session.
Thank you.
Yeah,
Thank you.
shall we go ahead with the formal vote of thanks, Jenny? If no question.
Okay, ma'am. Okay, ma'am. The smallest thanks is always worth more than the effort it takes to give it. Now I request Dr. Mary Shandi, Assistant Professor of Botany, to propose the vote of thanks.
Sustainable development requires human ingenuity. People are the most important resource. So your talk on clothes, which are the basic ingredient, is also one of the basic responsible factor behind the climate change. And a few of your slides, which gave insights into the topic, are how to enhance biomass production. The basic fundamental needs in science is missing, which has led to a gap. And then the metal regulation of iron and zinc. How to maximize yield, which requires a balanced plant nutrition, and how zinc dependent proteins get zinc. The session certainly benefited from your breadth of knowledge and depth of expertise. Your lecture definitely created a significant impact on every participant connected here. Thank you so much, sir, for your inspiring us to extraordinary heights, which will be very much useful in our career and future research. Thank you so much, sir, for your presence.
Thank you. Thank you very much. Hope.
Thank you, Kumaran sir. Really, you spent much valuable time with much preparation. We are very fortunate to have you as a resource person, and it's a proud moment for us. A person hailed from India and working at US and transferring knowledge to India. That's very great for us. We expect your future help in building our younger generation at India. Thank you so much.
Sure.
Thank you so
Thank you.
much.
Thank you very much. Thank you very much again,
We'll
Dr.
come.
Glory. Yeah, thank you very much, Dr. Shanti Sheelan, for your valuable vote of thanks. Definitely, I'm happy. I'm very glad to participate, and definitely it's my duty. Whatever the knowledge I know, I really love and like to transfer to our younger generation. Of course, it's my duty. It's my duty. It's because what we are facing is not a simple issue. We are thinking it's a simple issue, but definitely it's not a simple issue. It's we know, for example, COVID-19, we know, but this issue is unknown. It's going to affect soon. So definitely it's really a challenging issue we are going to face or we are facing. So it's a great initiative and a great participation. Thank you very much for the nice opportunity. And it's again, it was a great pleasure, and I'm happy to assist, or definitely we'll keep in touch. And thank you very much again. Excellent, great, nice. Thank you, thank you all.
Thank you. We do. It will. Thank you, sir. Thank you so much. Thank
Dear
you, sir.
Thank you. So
participants,
I can take off. Yeah, right. I can
Okay.
take off, right? Yep.
Yes,
Thank
sir.
you.
Yes, sir. Sure, sure, sir. Sure, sir.
Thank you. Have a great
Thank you.
day.
dear
Sure,
participants,
sure. Thank you.
dear participants, again we will meet today at 4:50 p.m.
Dana ma'am? Dana ma'am?
Shanti?
Can we end the meeting? Because only by evening
Yes,
the
yes,
meeting
we
will
will
be
end the
posted.
meeting.
How
Shanti,
we
we
will
will
end the
end the
meeting? Okay, ma'am.
Okay, okay.
Thank you, ma'am.
End the
Thank you.
meeting.
Bye. See you.